Hello, and welcome to the latest e-learning event from Petro Industry News, sponsored by Waters Corporation. My name is Tom Lynch, and I'm a consultant editor for Petro Industry News. Now, before we start, I'd just like to go over some housekeeping. On your display, you will be able to see a questions panel. Please type and submit any questions in this area, and we will review as many as we can at the end of the presentation with a dedicated question and answer session. Any questions which we cannot cover in this session will be answered by email after the event. If you have any questions or technical problems with viewing or hearing today's session, please use the questions box and we will respond to technical system questions instantly. Also, please make sure your internet speed is adequate and be aware that wireless connections may disrupt your session. We are making this e-learning event available for on-demand review, and this will be on our website in 24 hours time. Now, lubricants are used in almost every aspect of our lives, and efficient lubrication is important in keeping our world moving smoothly and efficiently. Modern lubricants are extremely complex mixtures of oils and additives, which have been specifically formulated for their intended application area. And until recently, the detailed compositional analysis of many lubricant formulations and additive components has not been a realistic option for many laboratories. Therefore, it has been difficult to generate chemical composition versus performance correlations for lubricant research, development, and optimization. Our speakers today are Dr. Eleanor Riches and Dr. Caitlin DaCosta from Waters Corporation, and they will be describing high-resolution mass spectrometry and statistical analysis approaches to lubricant formulation, research, and development. Eleanor is a principal scientist in the scientific operations team at Waters Corporation based in Wilmslow, UK. Eleanor joined Waters in 2008 and currently works on high-resolution mass spectrometry instruments with a particular focus on applications in the polymer, oil, and petroleum industries. Caitlin is a senior applications scientist for the UK and Ireland regions, covering pre- and post-sales application support for both high-resolution and tandem quadrupole mass spectrometers. Her focus is the chemicals and materials market and extractables and leachable applications. Prior to joining Waters, Caitlin completed a PhD focusing on the application of high-resolution mass spec and ion mobility to the analysis of lubricant oils. I have to admit, it will be nice to hear from Caitlin again, as I was her industrial supervisor for her PhD. So I'm very interested to hear the latest developments in this area. Without further ado, I pass you over to Eleanor and Caitlin to tell us more.
Thank you very much, Tom, for the kind introduction. Hello, and welcome to today's presentation. I'm going to start with an overview of the topics that we're going to cover today. So initially, I'm going to do a brief introduction to mass spectrometry, talking about what it is and what information we can gain from mass spectrometry. I'll then move on to talk about structure and property measurements, including commenting on what physical properties are typically measured for lubricant oils and how mass spectrometry could potentially underpin those physical measurements. I'll then move on to outline the experiments that we carried out, telling you how we acquired the data and what methodologies were used. And then I'll move on to give you a brief overview of the initial review of ion mobility data for the lubricant oil analysis we've done. And I'll show you how we can qualitatively make statistical comparisons between the lubricant oils we've analysed. I'll then hand over to my colleague, Caitlin, and she will tell you in more detail about the multivariate statistical analysis approach and how we can elucidate unknown species discovered in the lubricant oil samples. She will then also tell us a little bit about doing targeted screening for components of the lubricant oil and how we can go on to quantify the species that we've found. And we'll finish by doing a typical summary and conclusion. So, to start with a brief introduction to mass spectrometry. Now, I'm well aware there are likely to be many people on this call who are very familiar with mass spectrometry, but I'm also conscious that there might be a few who have not used the technique before. So I want to be sure that we're all comfortable in understanding what the technique is and what kind of data it can give us and what the data looks like when we go on to review it. So, what is mass spectrometry? Well, to put it simply, it can be considered as a way to measure or weigh molecules within a sample. And if we consider the types of samples we might encounter, they could be anything from a cosmetic cream to food and drink and the packaging that contains it, fine and specialty chemicals, or more relevant to this talk, lubricant oils or even crude oil. And each of these samples can be prepared and introduced into a mass spectrometer. All types of mass spectrometers typically comprise these four separate sections: the ability to ionize the molecules, to separate the ions, to detect the ions, and to report back to the user. Of course, the most important step is the ability to turn molecules into ions, because without ions they would not be detected by the mass spectrometer. And those of you who are familiar with the technique will be well aware that there are a plethora of different approaches that you could choose from. And I've shown just a small selection in this illustration here. And as you can see, it is a veritable alphabet soup of different acronyms. I've shown two in particular in larger font, and that's not a value judgment. I'm not saying they're better or more easy to use than any of the others. All I mean is that these are the two techniques we used when we did the analysis of the lubricant oils. We used electrospray ionization, or ESI, and something called an atmospheric solids analysis probe, or ASAP. Once we've generated ions, they need to be separated. And again, there are many different ways of bringing about this separation. But in the instrument that we use for the analysis that we're going to talk about later, there is a quadrupole, which, as the name suggests, is 4 poles or metal rods that have an electrical potential applied to them. And the ions are sorted according to their responses to those electrical potentials. There is also a time of flight, or TOF region. And in the TOF technique, ions enter onto a pusher, which again applies an electrical potential and sends the ions into a tube, or flight tube, under high vacuum. As you can see in this little cartoon, in many instances, the ions are sent into the tube and then turned round and sent back again, because the longer the flight tube, the better the resolution of the mass spectrometer, because the ions are sorted according to the time that they take to fly through that tube and reach the detector. The longer the flight time, the better the resolution, and we are doing high resolution mass spectrometry. So what information can we get from mass spectrometry? Well, as mentioned, we get data about the molecules within a sample in the form of ions, and the output is a mass spectrum. And we can see here an illustration of what a mass spectrum looks like, with mass to charge ratio, or M over Z, on the x-axis, and ion intensity on the y-axis, either as a relative percent or measured in absolute units. And this is an example of a real mass spectrum, which we will be discussing later in this talk. And what types of questions can mass spectrometry address or give us answers about? Well, typically, the first thing we might want to know is there a particular known compound in my sample? Or is X in my sample? And this would be described as targeted screening. If we found X in the sample, we might want to know how much of it is present. And this we would call quantitation or quantification. Then we might want to know what else is in our sample, So this might be described as non-targeted screening. And if we find other unexpected species, we might want to elucidate or understand the structure of those new found species. And finally, we might want to compare our sample with other types of samples. So obviously this would be described as comparison. And we might want to do this if we notice that something has changed in our formulation. Perhaps something has appeared or disappeared. Or maybe we want to compare our formulation with a competitor's formulation. For example, what are they adding that we're not adding? And mass spectrometry can address each of these points. Now I'll go on to give a few comments about the idea of structure and property measurements. I'm sure you're all very well aware of the types of physical properties that can be measured for lubricant oils. And when I was thinking about how to answer this question, what physical properties are typically measured, I decided to take an interesting article from Tribology and Lubrication Technology and pass it through a word cloud generator, the output of which you can see here. The article was entitled Component Performance in Engine Oil Formulation and looked at how engine oils are formulated and the tests they have to go through. And I think you'll agree this is a pretty fair representation of the types of physical property measurements you might make. Anything from viscosity or stability, shear or volatility, measuring specific additives, and understanding how these combined affect the wear and surface performance of a particular lubricant oil. And these properties can of course be measured by many different types of instrumentation. For example, rheometers, thermal gravimetric analysis, microcalorimetry, differential scanning calorimetry, oxidation stability, measurement of specific elemental components, foaming characteristics, cloud and pour point analyzers, and closed cup flash point analysis. So these are things that you're very familiar with. But how could mass spectrometry underpin or complement these physical measurements? Well, there was a really nice comment in the conclusion of the aforementioned article, where the author said that a good formulator would take a base oil plus additive package and aim to achieve synergistic performance results and avoid potential antagonistic effects. And I'm sure we can all agree that lubricant function depends on the stability of its chemistry and molecular architecture under the stress of the application. So we can start to see that the underlying molecular structure of the lubricant oil would be inevitably linked to these observed physical properties. And now we can see that mass spectrometry and the measure of the molecules within the lubricant oil can give us information about the observed physical properties. I'm now going to go on to tell you all a bit about the outline of the experiments that we carried out on a variety of lubricant oil samples. This initial work was a proof of principle, and it was completely blind. We merely took an off-the-shelf selection of domestic car engine lubricant oils, 5 different formulations, 2 of which were well-recognised premium brands, and 3 generic makes which were identified as being specifically formulated for different marks of car, all of which were described as 5W30 fully synthetic lubricant oils. It's a very simple sample preparation, as you can see here, with a slight difference depending on whether we were preparing the sample for electrospray analysis or ASAP analysis. The sample introduction was also slightly different for each technique, but in both cases we wanted to ensure that the overall approach was extremely quick and simple. For electrospray we used a 5 microliter loop injection, but I want to point out that there was no chromatographic separation involved. The entire analysis took a matter of 1 to 2 minutes. and the same was true for the ASAP analysis. Throughout this work, we're doing high-resolution mass spectrometry, and to ensure the best mass accuracy, we deploy a technique known as lock spray with the information shown here. The instrument that was used for all of our analyses was the SYNAPT XS HDMS ion mobility mass spectrometer. And as I indicated on earlier slides, we chose to use two different ionization approaches, electrospray in positive mode and atmospheric solid analysis probe, also in positive mode. And I'll explain why that is in a couple of slides' time. And the mass spectrometer itself is an ion mobility separation in tandem with high-resolution mass spectrometry. And because we're not using any upfront chromatographic separation, we chose to employ ion mobility to afford a degree of deconvolution and separation for these very complex lubricant oil formulations. As I've mentioned, two different ionization techniques were used. And this is because electrospray tends to favour more polar species, that is, those that contain nitrogen or oxygen atoms. This means that ionisation of the additives is favoured. When using ASAP, it can also ionise the additives, but it's also possible to ionise components of the base oil as well. So we get separate complementary pieces of information from the two different ionisation techniques. ASAP is a simple dilution and dip technique. Again, there is no chromatography and we get our data in a matter of minutes. If desired, we can apply a thermal boiling point profile, again to deliver a degree of separation and deconvolution for complex samples. Here we can see on the left the ASAP assembly. The larger black portion is seated on the top of the mass spectrometer, and the central part is used for sampling and insertion into the mass spectrometer. And you can see the ASAP sampling procedure in the three photographs in the middle. With dipping, the sample is deposited on a glass capillary, which is then inserted into the mass spectrometer. As mentioned, if we want, we can deploy a temperature ramp. And this is a description of the temperature ramp used in our analysis. We started with a setting of 150 degrees C and spent 30 seconds dipping into our sample. After 30 seconds, the holder with the sample was inserted and held still at 150 degrees for a further 30 seconds. At 1 minute, a value of 650 was input, and this started the temperature ramping upwards. It reached 650 and was held there until the acquisition finished after 3 minutes. Deploying this temperature ramp resulted in a thermal desorption profile as shown here. The black outline shows the sum total of the ions evolved at each temperature, or the total ion chromatogram. And here I overlay an approximation of the thermal ramp described in the table. While we can't actually record this kind of output, there is a displayed temperature change on the instrument page, and by observing this temperature readback, I was able to generate this curve. Now we're going to take a look at the data acquired using these techniques. First of all, a very simple overview, and then how we can make statistical comparisons between the different lubricant oils. As mentioned, two different ionization techniques were used, so let's take a look at the data we can get from each. Starting with an example of an electrospray mass spectrum, and we can see that the more intense species are predominantly additives from the lubricant oil. However, if we take a closer look at the higher mass region, we see some low intensity polymeric species, which we recognise as polydimethyl siloxane, PDMS. If we do a similar review of the ASAP mass spectrum, we can see that more intense species again are predominantly additives, and there are some common ions between the two, for example at M over Z 422. And this time, if we take a closer look at the higher mass region, we see some characteristic clusters that are representative of the base oil species. As we saw on an earlier slide, using a temperature ramp can generate this thermal desorption profile. And we're able to extract and observe the spectra acquired at each different temperature point. Here at A, on first insertion of the sample, we see many low mass species, some of which are related to the solvents involved, and some are low mass species from the lubricant oil. In region B, we start to see the evolution of some of the lower mass additives. And under region C, as the temperature starts to ramp upwards, we see the evolution of higher mass additive species. And as we saw previously, as the temperature gets higher under region D, we start to see components of the actual lubricant oil, base oil itself, being evolved. So we can get a degree of deconvolution and start to understand key components of our formulated lubricant oil. As I mentioned in the earlier slides, the mass spectrometer we use includes ion mobility. So what does ion mobility data look like? Well, I'm just going to show you a cartoon illustration so you can understand what we're seeing when we start to look at some real data. As you can see here, we typically have mass to charge on the x-axis, similar to a mass spectrum, but this time the y-axis shows drift time. That's the amount of time that the ions drift through the ion mobility region. If we have a small, low mass species, it will have a low mass to charge and it will have a short drift time because it's very mobile and moves quickly through the ion mobility region. If we have a heavier species, then it will have a higher mass to charge, and because it's larger, it will have a longer drift time as well. However, in the case of a species that is isomeric, it has the same formula and therefore the same mass to charge, but if it has a larger, more open structure, it would have a longer drift time than the smaller, more compact structure. So now we have an additional dimension of separation for our complex samples. Ion mobility enables us to separate species based on their size, their shape, and also their charge, which I haven't shown in this illustration. Ion mobility also enables us to measure a key physicochemical component of our individual ions, which is called collisional cross-section, or CCS. And this is a key identification point, which Caitlin will tell us more about later in the presentation. So what does real ion mobility data look like in DriftScope software? Here we can see the ion mobility data for the ASAP mass spectrum we saw a couple of slides earlier. And we see the same mass spectrum in the bottom image. The bright coloured image is illustration of the ion mobility data, again with mass to charge on the x-axis and drift time on the y-axis. As I mentioned, we see the smaller, more compact ions in the bottom left-hand corner, and the larger, less compact species towards the top right. In this image, the brightness indicates intensity. So M over Z 422, the most intense ion, is the bright yellow blob in the centre of the plume of colour. The other benefit of using ion mobility is we see that species are related, are defined and found in certain regions of the mobilogram. So for example here, I've highlighted species that are additives. We can also observe related species that belong to the base oil itself, and below the main plume we can see these regularly spaced clusters of ions that we know belong to a polymeric species. and this polymeric species is not easily observable by eye in the mass spectrum below. It is only by deploying ion mobility we can observe these different regions. Fundamentally, in this talk, we wanted to look at statistical analysis approaches to lubricant formulation research and development. So how can we qualitatively compare our ion mobility data? If we want to explore how this ion mobility data can be used to make comparisons between different lubricant oils, we can use HDMS Compare software. And this is the output from that software. In the top left corner, we see mirrored overlays of the thermal desorption profiles that I showed you in earlier slides. Anything that is coloured orange or shades of brown relates to lubricant oil sample 1. And anything that is coloured in shades of blue relates to lubricant oil sample 2. In the upper right quadrant, we can see sample information in a list format. And in the lower left region, importantly, we can see a visual display of the ion mobility data, which highlights the regions that have significantly different ions in each sample. So in this view, anything coloured orange is significantly different in sample 1, and anything coloured blue is significantly different in sample 2. So we can see immediately from this view that there are statistically significant differences between the two lubricant oil samples, the data from which we've processed in this particular example. Finally, we have the ability to place a zoom window on the visual display of the ion mobility data to allow us to investigate regions of interest in more detail. Now, we need to remember that this is a qualitative statistical analysis and comparison of our data. I'm now going to hand over to my colleague Caitlin, who's going to give us a more in-depth look at the multivariate statistical analysis approach and elucidation. Over to Caitlin.
The first approach builds upon what Eleanor has already described and involves a statistical evaluation of the data using a technique called multivariate analysis, or MVA. So the approach is outlined in this workflow. First, we need to acquire and then process our ion mobility high-resolution mass spectrometry data using the techniques we have already discussed. Once we have this data, we can then subject it to a multivariate analysis to identify markers that are significant to each sample and markers that account for the observed similarities and differences in the different lubricant oils. If we then want to understand these similarities or differences further, we can begin to elucidate the markers and identify what molecular species they relate to. Finally, we can confirm these identifications by reanalyzing standards to check our elucidation process is correct. Before we begin looking at the data, I just want to provide a brief introduction into multivariate analysis. Multivariate analysis, or MVA, is a collection of statistical techniques that are used to analyze datasets containing two or more variables that can occur simultaneously. If we take the following as an example, here we have an experiment containing 3 different sample sets. Each sample set contains replicate measurements, and there are at least 3 different variables. I think we can agree that the most significant and obvious variable is the fact that each sample set contains a different class of animal. What MVA will allow us to do is determine the relationship, so the similarities or differences between these sample sets at a component level, and then group the samples accordingly. As I mentioned, MVA is a collection of statistical techniques. So these techniques can either be unsupervised or supervised in their approach. Unsupervised methods are exploratory in nature, and they can help us identify possibly unknown patterns between different data sets. When data are inputted into an unsupervised analysis, we do not provide the statistical model with any additional information. Therefore, any separation of the samples is a direct result of differences in the individual components of different sample sets. Supervised models, on the other hand, require some form of input. Here, we're telling the model that we expect sample set A to be different to sample set B, for example, and we then use the model to identify the components that account for those differences. So, investigative analysis often uses an unsupervised approach in the first instance. But what is this and what information can it give us? Principal Component Analysis, or PCA, is an unsupervised method in which the number of variables in a dataset are reduced while preserving as much information as possible. The outputs are often in the forms of a scores or loadings plot, which we will discuss now. So a scores plot allows us to observe patterns and groupings between samples. Samples are things like the circles, squares and triangles, and those that are positioned closely together are positively related to each other in some way, while those that are far apart on the scores plot are negatively related. So if we take the previous example, we would expect the mice to cluster together in group 1, the monkeys to cluster together in group 2, and the dogs to cluster together in group 3. The loadings plot helps us to identify which variables will have the largest effect on each component, from which we can identify our marker ions. Markers in the centre are classed as having a weak influence and are often common throughout the different data sets. However, those off on a trajectory strongly influence the component and are responsible for the observed differences between different sample sets. So how can we use this for the analysis of lubricant oils? So here we have the results from our 5 different lubricant oil samples that Eleanor mentioned in the experimental section, with 5 replicate measurements of each lubricant oil. It's important to note here that the data I'm showing relates to our electrospray ionization approach. We can see on the scores plot the replicate injections cluster well together, showing good grouping and highlighting the reproducibility of the approach. We also see clear separation of the different lubricant oils, showing there are some statistically significant and measurable differences between the different oils at a molecular level. Moving to the loadings plot, we see 2 clear trajectories of markers. So we have a trajectory to the left, highlighted in red, relating to markers observed in our lubricant oil sample 2, so lubricant 2. And to the right, we have a trajectory of markers highlighted in purple, relating to our lubricant 1 sample. Once we have identified what markers are responsible for the differences between our lubricant oils, we can return this information into our unified processing software to investigate further. As a result of the experimental approach and analytical equipment used, each marker will have a unique combination of measured retention time, drift time, and accurate master charge. It is a combination of these measurements that are fundamental for us to understand the molecular composition of our marker species. The unified software also allows us to easily visualize marker response across sample and monitor trends. So the plots here show the response of 2 marker ions from our lubricant oil dataset. To the left, we see a marker uniquely observed in lubricant 1, and to the right we see a marker that is elevated in lubricant 2. Once we have characterized our marker ions, the next question we often ask ourselves is, what are they? And how can we use our HD MS⁻⁸ data to elucidate the unknown species? The elucidation process involves 3 steps. First is an elemental composition prediction, followed by a database search, and then finally a technique called fragment matching. I'm going to talk through each one of these steps in more detail, but before I do, I just wanted to recap on the HD MS⁻⁸ data acquisition mode. Here we have a schematic of a mass spectrometer similar to the one Eleanor described earlier on. We can see our analytes entering the mass spectrometer from the left, so following this blue line. And what's in the mass spectrometer are ions, which are represented as green and red dots, will pass through a series of ion guides, and a collision cell before entering the time of flight tube and reaching the detector. When we're acquiring data using HDMS to the E, what we do is rapidly alternate this collision cell between a low and a high energy. The low energy allows us to transmit our ions intact to generate our low energy accurate mass precursor ion spectra. The elevated or high energy induces fragmentation of our ions to generate our high energy fragment ion spectra, which allows us to get structural information. Because we can very rapidly alternate between this low and high energy, we can do this for all of the ions entering the mass spectrometer throughout an analysis, giving us a lot of additional information. So, back to our elucidation process. As I mentioned, the first step in the elucidation process is an elemental composition prediction using the accurate mass to charge of the precursor ion. So this is the precursor ion of our marker species. To do this, we use the high-resolution, accurate mass measurement of our unknown species in our low-energy spectrum and the associated isotope pattern that we observe. Together, those two combined allow us to predict an elemental composition. We have an example here for an unknown species with a mass to charge of 1194.818, which gives us a predicted elemental composition of C73H108012. Stage is to try and identify any potential compounds that have this proposed elemental composition. To do this, we typically conduct a database search. So this may be through an internal database or a specialized database, or in fact could just be searching ChemSpider. We enter our proposed elemental composition or our proposed formula into ChemSpider, and this returns a list or a series of proposed structures of known or common compounds that have that elemental formula. The final stage is a technique called fragment matching, which helps us narrow down our list of proposed compounds and increase confidence in assigning a proposed structure to our unknown marker. Here, we take the structure of the compound that we've identified through our database search and do an in silico fragmentation. So we theoretically break apart that molecule and see if we can identify or match any of the theoretical fragments with ions observed in our high energy spectrum. And this is what the results look like when they're combined together. So here we have our low and high energy data with our precursor ion on the top in our low energy spectrum and our fragment ions in our high energy spectrum. To the right, we have our results in a tabular form from our database search. So we have our experimentally derived precursor ion mass to charge, the proposed elemental composition with the IFIT confidence measurement for that. We then have the results of the database search, so we have 4 entries in that list of potential molecules that this could relate to. We have our fragment match results, so how many ions that we have observed in the high energy that we can relate to those proposed structures, and then some additional information such as the number of times this compound has been cited in the literature. And all of this together can help increase our confidence when we're looking to elucidate an unknown species and assign a potential molecular component to that. The next stage in this process would be to purchase and acquire standards to confirm our elucidation approach. The second approach I want to discuss is a targeted screening and quantification approach. So in the work that we've been talking about so far in this presentation, we've really been focusing around lubricant formulation and development and R&D, where we're using a non-targeted approach to really determine what is in the sample and is it important. Once we've identified those markers, so these could potentially be additives within our lubricant oil, we can use this information in a targeted manner for routine screening. When I'm talking about targeted screening, I'm talking about a well-defined list of target analytes where we can use selective or more selective acquisition and/or processing methods. And this can be both qualitative and quantitative in nature. The questions that we are typically asking when doing a targeted screening is firstly an identification question. So, are my expected additives present in the sample? The second question then, if we have identified our additives, is how much of the additive is present in that sample? So, quantification. When carrying out a targeted screening approach, the first thing we need to do is define an appropriate target list. So this is a list of expected additives or analytes that we want to screen against. The target list can be created in a variety of different ways. What I'm showing here is a Unify library entry that we are going to use to create a list to screen for. Each library entry contains a range of information about our target additive. So we can see here just in this example, we have the structure. From the structure we can get the formulae as well as the monoisotopic mass and the theoretical isotope pattern. We can also store a range of detection results against that library entry. So these include things like expected retention times, expected mass to charge, and expected fragments that we can use to increase the confidence in our identifications when we are screening. So what do the results look like, and how can we utilise our HDMS to the E data acquisition mode? Well, when we screen against a list of additives, we want to make our identifications using multiple identification criteria, which, when combined together, enhances our confidence in the presence of our expected analytes. So looking at this example, we have a list of identified targets with their observed mass to charge, the associated response, and any observed adducts. When doing the screening, we can look to our library, which has expected retention times, as I said, to compare the expected versus the observed retention time, as well as looking at things like the mass accuracy of our measurement. So is this within what we would expect for the instrument that we're using? We can also look at the isotope fit to make our identification, as well as looking at the high energy data and the number of expected or theoretical fragments that we can find. Finally, if we're using ion mobility, we can compare expected versus observed collisional cross-sectional, or CCS, measurements. So CCS is a measurement of an ion's size, shape and charge that is directly related to ion mobility drift time. Finally, once we've identified the presence of our target analytes, we may want to quantify them in our sample, so calculate the concentration that we observe in our different lubricant oils, for example. To do this, we would start by analysing a standard calibration curve over the concentration range we are expecting to see. We can then analyse our samples and use that standard calibration curve to calculate the concentration of our targeted additives in our sample. So now I'm just going to wrap up and provide a summary of what we've shown today, discuss some future work that we are undertaking, and then open the floor to questions. So the question that we really asked ourselves at the start of this was, how can high-resolution mass spectrometry and statistical analysis assist with lubricant formulation, research and development? Well, hopefully we have shown that typical lubricant formulation analysis often relies upon physical measurements. However, we can use mass spectrometry to supplement those physical measurements by interrogating the formulation at a molecular level. Our experiments demonstrate that ion mobility-enabled high-resolution mass spectrometry can be used to rapidly detect a range of lubricant additives and base oil components in off-the-shelf formulations. Combining the analytical approach with statistical analysis of the data can help us to identify unique oil markers that differentiate lubricant oils at a molecular level. And we can then use the data to elucidate these unknown species. Finally, the approach can also be used for routine screening and quantification. If you would like to find more information about the work we've shown today, we have a related applications note that can be found on the Waters website or online, as well as several posters that we've presented across different conferences. When it comes to what work we're planning to carry out next, really our work revolves around this statement, that lubricant function depends on the stability of its chemistry and molecular architecture under stress of the application. And what we're hoping to do is show that we can use a range of analytical techniques, including mass spectrometry, to really understand this structure-property relationship. Some of the immediate experiments we have planned is looking at degradation of different lubricant oils, and can we map that degradation from both a physical and molecular perspective. If you would like to hear more about this structure-property relationship, my colleague Eleanor is presenting a talk at STLE in May this year, titled Analytical Approaches to Chemical Structure and Physical Property Measurements of Lubricant Oils, where you can hear more about the work we've done and the work that we are planning to do in the future. So I just wanted to finish by saying thank you to you all for taking your time to listen to our work today. I also want to thank Tom Lynch for being a moderator and for his kind introduction. And thank you to Petro Industry News for hosting this webinar. And I'd like to open the floor to any questions. Thank you very much.
Well, thank you, Eleanor and Caitlin, for that fantastic insight into developments in high-res mass spec for lubricant analysis. In my view, this is the most exciting emerging technology for the analysis of complex formulated products such as lubes and fuels, and particularly for the elucidation of structure-property relationships to understand which chemistries really contribute most to performance enhancement in real life. That understanding allows the optimization of performance versus cost, and also development in new additive chemistries to improve performance in critical areas. Now again, to remind the audience, the presentation will be available at Petro Online in the next 24 hours for on-demand viewing. So if you missed any points of particular interest or had any sound or vision issues during the presentation, you can revisit it at your leisure. You will see a poll pop up soon. We would very much like if you could answer any of the questions there as we go through our Q&A session, which is coming up now, and we'll now have a look at some of the questions submitted by the audience. Now, our first question is around ionization techniques. You mentioned two ionization techniques in the experimental plan. Why didn't you just use electron impact ionization, as that is the most common?
Thank you very much, Tom. And thank you everyone for joining us today. And that's actually a great question to kick off with. This is Eleanor, and so I'm happy to take that question. In the examples that we showed, we used either electrospray or this technique called ASAP, Atmospheric Solids Analysis Probe. And the key thinking behind that was twofold, really. Primarily to be able to address different components within these complex mixtures. Electron impact, or EI, is a popular technique and is widely used with, for example, GCMS, so it may be familiar to a lot of you. But it's quite an energetic technique and is carried out under vacuum. So there are some instrumental limitations or challenges using EI, so the source has to be under vacuum. Both the techniques we used in this presentation are atmospheric pressure techniques, so they're really easy to couple, to access our sample and to give us really quick and simple data outputs. As you saw, we were acquiring our data within 1 to 3 minutes, and we're able to choose what aspects of the complex mixture we focused on. So electrospray is particularly good for ionizing polar additives. ASAP gave us access to the additives and some components of the base soil as well. And with being at atmospheric pressure, we didn't have to be concerned with maintaining the vacuum and so on. So it was really for simplicity, working at atmospheric pressure, and for speed of acquisition in terms of keeping it to 1 to 3 minutes, and also having the toolkits available to address different aspects of the complex mixture. I think those are the key points, really.
Okay, thank you, Eleanor. We had a question here. Why wasn't chromatography utilised in this work to help separate the samples prior to mass spectrometry analysis?
Hi Tom, hi everyone. Thank you for the questions. It's Caitlin here. So yeah, I can try and answer that one. I mean, the reason that we didn't include a chromatographic separation in this analysis was really to reduce the sample analysis time, to allow us to generate our results very rapidly. It also, to a certain extent, reduces the complexity of the analysis by being able to directly introduce our sample into the mass spectrometer. So for both the electrospray ionization and ASAP approaches, our acquisition time was less than 3 minutes, which compared to 20, 30, or even over an hour, it's a lot quicker than what we would use for a typical chromatographic separation. What we hope we've shown is that we can still generate information-rich data without chromatography, using the alternative approaches such as ion mobility and techniques such as HD-MS. So I think, as Eleanor just mentioned and as we've shown, ion mobility is an orthogonal separation technique to chromatography, and it provides a really quick gas phase separation of our ions. So this occurs in the millisecond timescale compared to minutes versus chromatography. And ion mobility can help improve spectral clarity, it can provide structural information, and also be used as a unique identifier for screening. HD-MS enables us to simultaneously generate our precursor and fragment ion data for all of the ions entering the mass spectrometer without any pre-selection or additional separation. So as such, we can obtain our MS-MS data for structural elucidation for both targeted and unknown species. So even without chromatography, we're able to answer lots of questions about the molecular construct of our sample.
Okay, thank you, Caitlin. And again, something around the ionization front. We have a question here. You mentioned about the ASAP ionization for base oil characterization. Could you please give more details on the ionization mechanisms for the base oil?
Yeah, sure. It's another interesting question, and I guess I didn't have enough time in the main presentation to expand on the technique of ASAP, but so it's fundamentally based on classical atmospheric pressure chemical ionization, or APCI. So it's a gas phase ion molecule ionization mechanism approach. That's what makes it completely different from electrospray, where primarily the ions are formed in solution and then sprayed. So APCI type reaction mechanisms or ionization mechanisms are particularly good for more non-polar species, for example, the base oil in this case, because the processes involve electrons, high-energy electrons streaming from a corona discharge pin, and those electrons are in the gas phase, and they can initiate cascades of molecules, either involving atmospheric species, such as water that's present in the atmosphere, or nitrogen, other atmospheric components. And then as they form, they initiate a cascade of further transfer of charge, and ultimately the components of interest then become charged, the analytes that we want to look at, and those then can be detected by the mass spectrometer. So in the case of base oil, which is obviously hydrocarbon, there's no heteroatoms, or there's typically no heteroatoms present that are amenable to ion formation by electrospray. However, those hydrocarbons can have charge transferred to them by collision in the gas phase with other charged entities. And then once they pick up the charge, they can be detected by the mass spectrometer. I hope that goes some way to answering the question.
Yep, I think that was a great explanation there. I think Caitlin spoke a bit about IMS, and there's a question here saying, I've never seen IMS before. Can you explain a bit more about why it's useful? Why do you need it when you have a high-resolution mass spectrometer working at the front end?
Yeah, I'm happy to expand on the ion mobility bit as well, and I'm sure Caitlin can add in some thoughts too. So I guess the main power of ion mobility is it gives us this orthogonal separating power, and fundamentally it's based, as I mentioned in the presentation, on the size and the shape of the species. So particularly, as I understand it, in making novel additives, let's take that as an example, they can have different structures, and some additives can be polymeric and therefore can have perhaps branching features that give them particular functionality. And ion mobility helps us interrogate those structures, and that is not normally accessible by mass separation alone. Things can have the same mass but different structures and therefore different functionality. So that's one of the most powerful features of incorporating ion mobility. The other thing that Caitlin alluded to as well is it affords another dimension of separation. So obviously we've got mass resolution, and then we've also got this ion mobility resolution. So it gives us a greater system resolution without the need for the chromatographic separation, which is typically or often coupled with just high resolution mass spectrometry. So high resolution mass spectrometry and high performance ion mobility separation gives us this really good system resolution. And it also gives us the ability to measure collisional cross-section, or CCS, and that's a fixed physicochemical property of an ion, and therefore it can be an identifier. So again, if we've got screening of a complex mixture and we want to be sure that we're identifying key components, measuring the CCS can tell us for sure if we've got the expected species. So we feel that it gives us a whole raft of different benefits particularly in the world of analysing highly complex mixtures.
Okay, thank you. We're coming almost to the end of our time. We can maybe squeeze one more in. When would you do untargeted screening if you already know the ingredients in a formulation?
Hi, so I'll try and keep this one nice and quick to wrap up. But I mean, there are many reasons why you might want to carry out an untargeted analysis, even if you do know what the ingredients of the formulation are. And high resolution mass spectrometry can help you answer sort of lots of questions about a sample. So if you already know the formulation, you may want to look at changes in the molecular composition of a lubricant following use or stress. And this can help you understand the performance of different additives. So are they degrading? Are they reacting? Are they oxidizing or forming nitrates, for example? So you can really understand the chemistry of the product by looking at it using high-resolution mass spectrometry at a molecular level. And this can really help when optimizing formulations and increasing the efficiency of that optimization process. Alternatively, you may be looking at formulations where you're not sure what the chemical composition is, and you want to understand the product in a little bit more detail. So you could use it for things like tracking counterfeit products or investigating contamination issues. It can be used to look at variability between batches of lubricants or additives, or even understanding competitor products at a molecular level. So there's a lot of flexibility and a wide range of information that you can obtain.
Okay, thank you for that. I think we're just over the hour now, so we'll have to bring it to a close. But just like to say thank you all for attending this e-learning session brought to you by Petro Industry News and sponsored by Waters. Thank you to Eleanor and Caitlin for sharing their knowledge and experiences and answering the questions we've got them from the audience. Any questions we have not covered in the live Q&A will be answered by email shortly. Please also feel free to request your certificate of attendance, which you can see on your screen is available for download. Also, look out for the next e-learning sessions in 2021 by visiting petroonline.com and click the e-learning tab at the top of the page to see the details of the future sessions we are running, and more importantly, to gain access to all the past e-learning events. Until then, have a good day. From me, the team at Waters, and all at Petro Industry News team, stay safe. Thank you. Goodbye.